Alright, so now that we have Anaconda working and installed, we can just go ahead and type in Python and then type in the name of segment and track.py. You can type out the full name, or you can just type segment and hit tab and it'll automatically fill in the rest, which is kind of useful. And go ahead and hit enter and it'll start the script. So these are going to be a bunch of prompts asking you what you want to be doing. There's a lot of options here you can say yes or no to, so I'll try to go through those pretty slowly. First, well, I'll just answer the first 5 and then go through each of them. We're going to just say yes to everything because I need to show you guys how to run everything. 6 questions. So, do you wish to align the images? This is basically just because the images, the original images have a shift. You can sort of see it here, where the whole image register is moving. And that's a problem when we're trying to track cells because we need to be able to identify a cell location between different images. So we need to align the images. If you have a series of images where there's not a change in register like that, you can go ahead and skip this step. It's not that important, but it is super important to have aligned images. So you can follow the same cell through multiple images. So that's why we have that there. It uses a fast Fourier transformation to align the images, and I'll get a little bit more into detail in a minute. If you want to train or apply a classifier, this is basically just segmenting the cells. We need to create a black and white mask in order to track the cells, and segmenting or classifying them is how we do that. So since we don't have those black and white masks, we're going to say yes. Do you wish to track the cells, which is sort of the whole purpose of this script? So we're going to say yes to that because we would like to track the cells. Do you wish to output CSV files detailing data for individual lineages? So when we track the cells, we're not only tracking each cell, but we're also keeping track of ancestry, which I call lineages. And you'll only get this question if you agree to track the cells, or if you have a file that gets output when you track the cells already in the folder. So we're going to say yes to that too, just to show you guys how it works. And then, do you wish to analyze the images? And this is basically analyzing whole image fluorescence, so rather than individual cells, which is what gets analyzed when you track them, we're just analyzing the whole image. And even if you don't have fluorescence, because this process does work without any fluorescence whatsoever, but even if you don't have fluorescence, you need to say yes to this if you want to make videos. And then, do you want to make videos? It'll output a video with your data, and if you have fluorescence, it'll false color the image green, which you can change in the hard coding, but for now we just use green. And I'm going to say yes to that too. So once we go ahead and answer yes to everything, it'll tell me what my current working directory is, which is the cell tracking master. This is super important because almost everything you input when you're trying to tell it where a directory is relative to the working directory. So, for example, practice would be the working directory practice, and if I wanted to identify something within practice, I would have to put a dash, slash, a backslash at the end of practice, and then the name of the file within practice. And I'll show you how that works. So, now it's going to take us through the prompts that are needed for a couple of different things. It only asks you for information it needs depending on your answers to those first 6 questions there. So first it needs to know where all your images are, which is why we put them inside our working directory. So they're all in a folder called practice, and the script always checks to see if a directory exists. So if I were to put in a typo, it's not going to find the directory. But when I type it in correctly, it finds it and accepts it and moves on to the next part of the script. Now we need to know the first frame in the data set. That means the first phase image, which is going to be 448. And now we need the last frame in the data set, which is going to be 467. And the time per minute in frames, so this particular data set was taken every 30 seconds, which is every half minute or 0.5. And that's used for calculating time, essentially. And then the number of the first frame with the fluorescent first frame of the fluorescence image. So in this case, it's 449. If you don't have any fluorescence images, if you're just trying to track things, and then go ahead and type in 0. The tracking will still work. It just won't output any fluorescence data because it won't find any since you put 0. And then the number of frames between fluorescence images. So you can see here we took 449 and then 459, so it's every 10 frames. And that's just to prevent bleaching the fluorescence. If you take fluorescence images too often, you'll bleach the cells, and then you won't see any fluorescence, and that's a problem. So we take fluorescence images much less often than phase images. Now we enter the image file name preceding the channel and file number. So like I was saying, you need to have that -G or -P, -3 numbers and .tiff. So this is everything preceding that first dash. So in our case, it's 2017 12 book. And then this is asking for the name of the fluorescence channel. This is for if it's labeling a graph, which I don't think I actually have output, but it is a problem. An option, not a problem. Oh, and it's used to label the CSV file. Super important.